Hi, I'm Yvonne Doyle, and today I'm going to be talking about my project, which is developing and validating assays to design a stability study to extend the shelf life of monoclonal antibodies, also known as MABs. So a bit about me. So I graduated from Glasgow, Caledonia with a BSc with honours in chemistry with IT and instrumentation. I then moved to the University of Strathclyde and gained my Masters in Science in Pharmaceutical Analysis. I then went on to a pharmaceutical company and worked there for 22 years, with the last 10 years leading a stability team to extend shelf life of many antibody drug conjugates. In 2022, I decided it was time for a change, so I started to look for some jobs. I wanted to kind of stay in the same environment, cancer research, but I also knew I didn't want to work for a pharmaceutical company any longer, so I looked for jobs in NHS and academia. And the first job that I seen posted was this KTP. So here we are today. So project overview. The overall aim of this project was to develop and validate stability indicating assays which will be used to design robust stability studies to extend the in-use stability for monoclonal antibodies. The collaboration is between the University of Strathclyde and Target Healthcare, who are a wholesale pharmacy who supply generic, branded, specialised and unlicensed pharmaceutical products, which include MABs. The main issue for Target is the in-use shelf life of these MABs. For the majority of them, they have 24 to 48 hours at refrigerated conditions, which means a very small window for Target to supply these to clinics. So what is a MAB? A MAB is a monoclonal antibody, and it's a man-made protein that acts like human antibodies in the immune system. MABs are used as a type of targeted drug therapy for cancer to replace chemotherapy. It can be used in organ transplant rejection, inflammatory and autoimmune disorders, or other infections. They can also be used as probes to detect pregnancy and rapid diagnosis of hepatitis, influenza, streptococcal, and more recently, COVID-19. The World Health Organization are responsible for the naming of MABs, and they've reported that over 800 MAB names have been provided. As of June 2022, 162 MABs have been approved by at least one regulatory agency in the world. So how do they work? For this, I've put in an example of a cancer cell. So in the middle, you can see the cancer cell, and around the cancer cell are the proteins which enable the cancer cell to grow. The monoclonal antibody is a Y structure, and this works by locking onto the protein, and either dependent on the purpose of the antibody, it will either stop the growth of the cancer cell or it will kill it entirely. So the MAB that Target has chosen to design a stability study for is daratumumab, commercially known as Darzalex subcutaneous. Daratumumab is used as a targeted treatment against multiple myeloma, which is a type of bone marrow cancer. The current shelf life of the unopened vial of daratumumab is 2 years at 2 to 8. The current in-use stability is 24 hours at 2 to 8 and room temperature of up to 15 hours. Target transfers this product aseptically once it's opened into syringes so they're ready for use in clinic, and it's for this reason that the supplier of vials do not provide longer stability for in-use, as they have no control over how the customer handles these vials once they have been supplied. So where do we start when we're trying to design a stability study? We have to determine how a MAB shows signs of degradation. The diagram below shows 4 polypeptide chains. It's a very simple diagram. There is a more complex degradation pathway which I could have shown, but I just wanted to simplify things for everyone. The 4 polypeptide chains consist of 2 heavy chains and 2 light chains. There's a constant region which is constant for every antibody. However, there's a variable region which changes between 164 MABs that are on the market. They're very highly sensitive molecules because of proteins. As I said before, the diagram is a simplified version of the MAB structure, and there are more complex degradation pathways. If we were to analyse every degradation pathway, we would need a vast amount of money, time and personnel. So we've had to determine the best assays which will show degradation over time. So where do we start? The simplest way to start is look at the appearance, which is why appearance is the first test in a stability study. So on the left hand side would be your syringe at the start of the study, which would be a clear liquid. And then 14 days later, if your liquid has changed to orange, that is a clear indication that there's something wrong and the sample is not stable. The second would be pH. So the pH of the solution would be taken at the start of the study, and for this instance, it's pH 4. So this is showing that the sample is slightly acidic. If over time the sample becomes more neutral or more basic, then we know that there's an issue and that the sample is not stable. Next we have subvisible particulates. This is a test that will show us any particulates present that are not visible to the naked eye. So any particulates less than 50 micromolar can be seen through this subvisible test. We analysed 25 micromolar and 10 micromolar, and there's a certain specification. So then we have SEC HPLC. High performance liquid chromatography is an analytical technique, and there's many different high performance liquid chromatography techniques. However, as I said before, there's that many that we can't cover all with the budget and the cost and the time. So we have size exclusion chromatography HPLC, which is an analytical technique that separates a sample by size based on the elution from column filled with a porous gel. So this can identify any aggregates that are formed over time. The chromatogram below shows the monomer, which is the entire Y structure, and then you have your high molecular weight and your low molecular weight species. So we would monitor this chromatogram over time, and we would see if there's any additional peaks that are formed which would hint at any degradation, which would show aggregates. So this is one of the main tests that we monitor over time to ensure that there's no additional peaks, or if there is any additional peaks at a low level. Next we have capillary electrophoresis. This uses non-reduced, which monitors the purity of the denatured intact MABs, and reduced, which monitors the light chain, heavy chain, and non-glycosylated heavy chain. The CE system can also use charge isoelectric focusing to characterise the sample and ensure no significant changes. The electropherogram below shows the reduced and the non-reduced. So at this number 1, we have the internal standard. Number 2, we have a light chain. Number 3, we have the non-glycosylated heavy chain. Number 4 shows the heavy chain, and number 6. Number 7 shows 2 heavy chains and 1 light chain. Number 8 is another non-glycosylated heavy chain, and number 9 is the entire structure, which is classed as a monomer. So again, if we see any signs of any extra peaks through time, then that shows signs of degradation, and we can say have to investigate. Next is ELISA. ELISA ensures the biological aspects of the MAB are not affected over time. So it's not looking at the structure, but it's looking at the relative potency of the solution over time. ELISA has been the most tricky part of this because I have no previous knowledge of it. So this is one thing that I've had to do a lot of research on, and there's many different steps, there's many different variables, which has caused a lot of issues for myself. So we have many steps. We have the coating step, which is you're coating the target antigen onto a plate, a 96 well plate. You incubate that overnight, and then next day you block it. So you're blocking it so that no interference between any other antigens or anything else will come up on the plate. They then capture the analyte, which is the primary antibody, and then you add a secondary antibody, which is detection, and then you add a substrate which will change colour, so you'll get a colour change from blue. You incubate that for 10 minutes, and then you stop the solution with a sulfuric acid, which then turns the colour to yellow. Then you then have your analysis and calculation. Because of the nature of this assay, it's not very consistent. It's not anything can affect it. So time, if you incubate for 5 minutes, 10 minutes longer, then it can affect the results. If you leave your substrate on 2 minutes longer than it should be, it can affect your results. So there's a lot of variables in this that has been very tricky. So method development. Once all the methods have been established, each one had to be developed. So most assays like the HPLC and the CESTS have standard methods for MABs. However, as each MAB is individual, there requires to be some optimization around sample concentrations, incubation times, etc. ELISA, as I said before, was the most challenging assay to develop due to the biological nature of the assay and the many individual steps and variables involved. The appearance, pH and subvisible particulate testing are all compendial methods, which means there's already methods published, and development is not required for these, only method verification. Method validation. So method validation is required to ensure that each developed assay is fit for purpose. So this requires analysing the following aspects of each assay. The selectivity or specificity, which is the ability to measure accurately an analyte in the presence of interference. Precision, which measures the values and how close they are to one another. Accuracy, which is how close the results are to a true measurement. The linearity, which establishes how linear your standard curve is. The range, which is generally the concentration range at which the linearity test is done. The stability, which is a critical part of this because it establishes how long you can leave a sample sitting prior to analysis. For example, if you start your experiment and then get called away for whatever reason, and you come back a few hours later, has your sample been affected, or can you just go ahead with the analysis? That tells us a lot. The limit of detection and limit of quantification. The limit of detection, LOD, is the lowest concentration at which the instrument can detect but not quantify, and the limit of quantification, LOQ, is the lowest concentration at which instrument can detect and quantify. So progress to date. So we've completed the appearance, pH and subvisible particulates method verification. We have completed the size exclusion, HPLC method development and validation, and we've had very promising results from that. We have completed the ELISA method development, but unfortunately the validation failed, so we have had to go back to redevelopment, and that's currently being finalised. We have completed the capillary electrophoresis SDS, non-reduced, reduced and CIEF method development, and we are due to start the method validation. We have targeted to initiate the stability study for daratumumab, but because of the delays, this is going to go into the week the project is due to end. So the key instrument, the capillary electrophoresis, was only purchased when I started the KTP, and it took a year to be installed and training rolled out, and it has been hit with errors, which has seen long periods of time where it was out of use. This has caused delays to the project, which means the study won't be initiated until the week the project ends. So plans for the future. As the project was delayed, the stability study has not yet been initiated. An agreement has been made between the university and Target that I can continue to use the university's kit to see the study through to completion. This means my employment continues with Target, who are looking to take this design in-house and extend the many more MABs that are on the market and their in-house shelf life. Significant achievements for me throughout this project have been completing my Prince 2 project management course and gaining my qualifications on that. Gaining knowledge on techniques like ELISA I had never used before. Providing Target with initial stability data and short-term stability, which allows them to answer queries from customers which they previously didn't have. For example, if they've left a vial out of a fridge and do they have to throw the vial out or can they use it? Do they have data on that now? And just gaining new confidence in my abilities. So thank you. Thanks for your time.